Applied research, a critical review. Applied research has various definitions which are usually dictated by the funding agencies. But in general, it's a research that seeks to solve practical problems. It includes novel ideas and industry partnership. What you see in this diagram is a framework for applied research in our biotech program at ABES. We offer three interrelated research courses. The first, BI 207 or structured research. Then the students move on to the independent research where they get the basic research skills. Some of them would be qualified as applied research, but mostly students will be repeating the previous papers. In the next level, advanced project or BI 303, where the students will try to incorporate some elements of applied research. Some of them may qualify as capstone projects. These will be competing in the technology fair. I was a bit curious to see the documentation on the applied research and did a keyword search on our Centennial website. Surprisingly, applied research and innovation resulted 10 times higher search research compared to ABES and biotechnology, resulting in an inverted pyramid. Although biotechnology and applied research are a part of ABES, the higher number of research for applied research means the college commitment to applied research and overarching aim. This is also consistent with Centennial's research progress. In 2015, Centennial hit the headlines by breaking into the first 10 research colleges in Canada. In 2016, we moved a step further. This is our current update on the research progress. We are continuously moving forward in the research rank for 3 consecutive years, and that indicates our commitment to applied research. And these research positions are determined by a number of factors such as research partnership, completed research project, paid student researchers, research income, and research intensity. I'll take you on a virtual tour to project laboratory. In a typical class, there will be 12 different projects, different topics running at the same time. Advising them, directing to the right resources, and doing the right biohazard management is always challenging. What you see here is live bacteria which was recorded in our modern microscope. In a typical microbiology project course, students undertake 10 weeks of intensive research and submit 3 formal scientific reports. This is the first time they are exposed to independent research and problem-based learning. On top of it, we also introduce applied research concepts and innovations. As a reflective practice, I sometimes question, do we overload them?
So warm. Wait a minute. How's that?
Nice and toasty all the way around.
Oh, hey, what's wrong? You hungry? I found your food.
But at the end, these are the skills that they can sell out in the competitive job market. At the start, the term innovation sounds scary to many students. I simplify using some good examples. Innovation doesn't necessarily need to be so complicated. We use Durham tube in our regular microbiology practicals. So this is an inverted tube inside a bigger tube invented by British bacteriologist Durham. The idea is so simple and anyone can get the recognition by a very simple idea. One way we can evaluate their skills needed for the applied research is through a smart test. We have transformed into smart technology, smartphone, smart screen, and why not smart test. This is a completely different topic. I make it simple. SMART stand for safety, mathematics, analytical skills, research and planning, and teamwork. This is not only an evaluation method, but also building the skills between the groups. So I usually divide them into group and exchange the skills. So this is also considered as a peer assisted learning. We have discussed applied research in general. Now let's talk about some examples. Nowadays, many faculty are involved with applied research and capstone project. I have limited to a few examples due to the time restriction. Safety first. All the colleges and universities working in the level 2 biohazard containment face some problems with the management of lab coat. Research is underway to build a smart lab coat. Interdisciplinarity is always welcome in the research. And how about bringing electronics into microbiology? I use the term microtronics, microbiology and electronics combined. And two students are involved with 3D printed devices in microbiology. Providing accommodation, there are certain students who cannot see the colors properly or color vision deficiency. So we are trying to provide a technological solution to this. Nanotechnology is now used in many fields, and there are two students, two projects which are looking into the nanotechnology based encapsulation, which I will be discussing later. This project is funded by the Applied Research and Innovation Center of Centennial College. Accommodation for color vision deficiency in microbiology. So this is a novel study investigating the different options for colorblind students. The colorblind deficiency or CVD is an inherited visual impairment that affects 1 in 12 men and 1 in 200 women. This is our college logo. The predominant color is green or green related. In the color vision deficiency, the most common type is red-green color blindness. Those individuals with this CVD may see this differently than we see. This is just an imaginary picture to show how blurry it would be when an individual affected by CVD look at this logo. You may ask, how does CVD or color vision deficiency play a role in microbiology? In microbiology, one of the basic techniques is gram staining, which was invented by Christian Gram in 1850s. To reach out the wider audience, I have excluded the technical details. To give you an idea, this is a very similar to hand washing techniques, a set of protocols. At the end of the gram staining, gram-positive bacteria can be differentiated from gram-negative based on cell wall composition. At the time of Christian Gram, this was a key information. The way we differentiate these two bacteria are based on colors. Gram-positive bacteria will be stained purple, while gram-negative bacteria will be stained pink. These are not really contrasting colors, and those with color vision deficiency will have difficulties in differentiating these. As a reflective practice, I wanted to see how students performed gram stainings effectively in their practical exam. I looked at my 3 years of exam papers and my notes, and I got an amazing finding which was presented in our annual session in 2017. These are the results. 14% of the students recorded wrong gram ID, although they did the gram staining perfectly. I won't say all 14% would be related to color vision deficiency. It's mainly the microscopic techniques, but color vision deficiency may play a role. In the long run, I identify at least 5 to 6 students who had some degree of color vision deficiency and had difficulty in differentiating pink from purple. I looked at some of the alternative to this issue. University of Tokyo developed an app called What Color app, which is freely downloadable. By moving the cursor around the color, it can easily tell the name of the color. But the problem is there are hundreds of different colors and how to classify them, purple versus pink. Later on, I developed a technique which is based on the viscosity of lysed cells, which can almost like differentiate gram negative from gram positive within 1 to 2 minutes. This is another novel research aiming to develop a smart lab coat. In Level 2 biohazard facility, lab coats are an essential PPE or personal protective equipment without which we are not supposed to enter the lab. But we don't have strict rules regulating in the management of lab coat outside. We can even students sometimes walking along the cafeteria. This is a prototype for a smart lab coat. When we exit the lab coat can be folded inside this inbuilt pouch. We hope to design this pouch with antimicrobial fabric. Currently, there are 2 projects underway using nanotechnology to build this fabric. To give you an idea, the nanoparticles are 1,000 times smaller than the microbes and have been proved effective in drug delivery and antimicrobial effectiveness. If you want further information, or if you are interested in contributing any of this applied research, you can contact my email. Thank you.